No, it's not glitch. I'm just messing with you guys. Okay, let's move on to the next slide. Okay. All right, so the next of these functional groups that we're going to talk about are carbonyls. Carbonyls are a carbon double bonded to an oxygen. Now, depending on what's on either side of that double bonded carbon, it could either be a ketone or an aldehyde. It's a ketone if it's surrounded by other functional groups or other hydrocarbons. It's an aldehyde if it's kind of just hanging off the end there with just one hydrogen. Now, acetone is probably the simplest ketone, and it's a popular and effective solvent that you guys might have heard of. Probably know it by its more common name, nail polish remover. Aldehydes, on the other hand, are often associated with very aromatic things like essential oils. For example, the two different aldehyde compounds that you can find are, well, two of the aldehydes you can find are cinnamaldehyde and vanillin. Any guess as to what smells are associated with those two aldehydes? It's not oranges, I'll say that. Anyway, now carboxyl groups are composed of a carbon double bonded to one oxygen on one side and then single bonded to another oxygen on the other side, and that oxygen is subsequently bonded to a hydrogen. Now, these are polar and hydrophilic, and they're considered an acid. Why? Well, they easily release that hydrogen ion. So, and when you release hydrogen ions, you're considered an acid. Now, carboxyls are a major component of amino acids that make up proteins. We'll get to that in a second. Now, real quick, the non-ionized form of a carboxyl is called an acetic acid. Okay? But often you'll see an ionized form, which is COO negative, and we call this carboxylic acid. Okay, so I mentioned that these are one part of an amino acid that you can see here. So what's this other part of an amino acid called? An amine, right? So we have the acid component, and now we have the amino component. Anyway, now amines are kind of the opposite of carboxyls because amines happily take up an extra hydrogen. And because they readily take them up, they give off OH in water. So it's considered a base. These are also considered polar and hydrophilic. So you'll see them in both their non-iodized form and also in their ionized form, their NH3 form, pretty often in cells. Okay, now sulfhydryl groups, another term for sulfhydryls are thiols, and they're often involved in protein structures. Okay, that's kind of their primary role, or one of their primary roles. And what they do is they actually form what are called cross-links or sulfhydryl bridges, where two sulfhydryl groups kind of on different parts of a protein will form a bridge together. Okay. Really quick, thiols are also polar and hydrophilic, as you can see. Okay. Right, so you form the cross bridges. Cool, covered that. Oh, okay. Fun facts about thiols. They're all often associated with really strong, nasty odors. For example, skunk spray has a lot of thiols in it. Cool. Okay. Now we're on to phosphate groups, also known as organic phosphate. They're polar, hydrophilic. They're also acidic. Now, important thing to note about phosphate groups here, notice how they have two negative oxygens kind of hanging off the end here. So this means they often contribute a pretty significant negative charge to whatever the hydrocarbon is that they're attached to. These get involved in phospholipids, and they're also very much involved in nucleic acids, right? Not too surprising, because they are acids themselves, so they kind of add the acid part to the nucleic acids. Now, nucleic acids you might be more familiar with are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Okay. Keep these guys in mind. They're really interesting, particularly when you start sticking chains of them together, say 3 of them in a row. Yeah, triphosphates. You'll see a lot more of these guys later, so keep them in mind. Think about what it means to have all that kind of negative charge locked together like that. Okay. Okay, so now we have methyl groups. Now we call them, a methyl hydrocarbon. It's kind of like the simplest hydrocarbon here, right? It's just a carbon with 3 hydrogens sticking off the end. Very simple. So it functions like you would expect a hydrocarbon to, like we originally talked about, right? So it's nonpolar. It's not hydrophilic, so it's hydrophobic. And it's neither an acid nor a base. So, as we kind of mentioned before, they can do all kinds of interesting stuff by adding these on. They can kind of change the function of sex hormones, like we talked about, testosterone and estradiol. They're also involved in how genes get expressed. And we'll talk a lot more about this later on in the course. But by adding a hydrophobic methyl group to the proteins that kind of help package DNA, the packaging of the DNA kind of moves away from the water and gets more tightly wound, making it very difficult or even impossible for those genes that are wound up to get expressed. We'll talk a lot more about that later on in the course, but this is when you kind of first see that now. So, yeah. So in lecture 3.2, we're going to be talking about biological macromolecules. So kind of what is a macromolecule to begin with. We're going to also talk about how you get macromolecules. And then the kind of 4 types of macromolecules that you find: carbohydrates, lipids, proteins, and nucleic acids. Just a little bit about nucleic acids. But before we get to 3.2, let's do our lecture question 3.1. What properties are shared between amino and hydroxyl groups? So figure out which of these is true. How can we answer this? What could we use to answer this question? Maybe something like this. Fill this out. See you guys in lecture 3.2.